This chapter brings us into the microscopic and macroscopic world of muscle tissue. By the end, you should be able to explain how the body organizes its three muscle types and what makes each one unique. We'll dive into skeletal, cardiac, and smooth muscle, looking closely at their structures, functions, and how they cooperate with tendons to create movement. You'll also understand the cellular mechanisms behind contraction and relaxation. And we'll touch on how muscle tissue develops and even regenerates. These objectives set the stage for everything from clinical applications, like how heart muscle differs from skeletal muscle, to everyday experiences, like why your arm can move a pencil with such precision. All muscle tissue is specialized for contraction, but not all muscle is alike. Skeletal muscle is voluntary and striated, allowing us to consciously move bones and control posture. Cardiac muscle is striated too, but it contracts involuntarily to pump blood rhythmically through the heart. Smooth muscle, on the other hand, is non-striated and involuntary, controlling the movement of food through your digestive tract or blood through your vessels without you ever having to think about it. Despite their differences, contraction and relaxation remain the unifying theme across all muscle types. The spotlight in this chapter is on skeletal muscle because it's the most accessible and widely studied, but the other types are equally essential to life. Muscle is defined by four essential properties. Excitability means muscle can respond to signals, usually from nerves, by generating electrical changes. Contractility is the ability to actively shorten and generate force, which is what makes movement possible. Extensibility allows muscle fibers to stretch without tearing, critical when opposing muscles pull in opposite directions. Finally, elasticity is the ability to snap back to their resting length after contraction or stretch, much like a rubber band. These properties together explain why muscle is unlike any other tissue. It can move us, resist forces, and adapt instantly to changing demands. All muscle tissue relies on the same contractile proteins, actin and myosin, but their organization changes everything. In skeletal and cardiac muscle, these proteins are arranged in repeating patterns, creating the striped or striated appearance you see under the microscope. Smooth muscle lacks this regularity, so it looks uniform or smooth instead. This difference in microscopic appearance isn't just cosmetic. It reflects the way the muscle contracts. Striated muscles contract with rapid, powerful force, while smooth muscle contracts slowly and in a more sustained fashion. Recognizing these structural cues under the microscope is one of the fastest ways to identify muscle type in histology. Skeletal muscle fibers are some of the largest cells in the human body. They are long, cylindrical, multinucleated, and their nuclei sit just under the cell membrane at the periphery. This structure results from the fusion of many embryonic cells, called myoblasts, during development. Skeletal muscle is under voluntary control, meaning we can consciously decide when to use it, whether to walk, write, or smile. It makes up about 40% of body weight and includes fibers that can be up to 16 inches long. Imagine a single cell spanning from your shoulder to your elbow. Skeletal muscle isn't only for movement, it also supports posture, stabilizes joints, and even controls sphincters, giving it both gross and fine control over the body. Cardiac muscle is unique because it exists only in the walls of the heart. The fibers are short, branched, and usually contain a single centrally located nucleus. A hallmark feature is the intercalated discs, which are specialized junctions that connect neighboring cells. These discs contain desmosomes, which act like rivets, holding the cells together during forceful contractions, and gap junctions, which act like doorways that allow ions to flow directly between cells. This electrical connectivity lets the heart beat in a coordinated rhythm. Cardiac muscle is also packed with mitochondria and myoglobin, giving it endurance to work non-stop from birth until death. Even though it's striated like skeletal muscle, it's not under conscious control. Its contractions are automatic, driven by pacemaker cells and regulated by the autonomic nervous system. Smooth muscle lines the hollow organs of the body, such as the stomach, bladder, intestines, and blood vessels. Its fibers are spindle-shaped, much shorter than skeletal muscle, and contain a single central nucleus. Unlike skeletal or cardiac muscle, smooth muscle lacks striations because actin and myosin are arranged more irregularly. This arrangement allows for contractions that are slower, sustained, and sometimes twisting, like a corkscrew. Smooth muscle is controlled involuntarily by the autonomic nervous system and can maintain tone for long periods without fatigue. Think of how blood vessels stay partially contracted to regulate blood pressure. Interestingly, smooth muscle also produces its own connective tissue, called the endomysium, which helps support and integrate it into organ walls. When comparing the three types of muscle, think in terms of structure, control, and function. Skeletal muscle fibers are long, multinucleated, and striated. They're under voluntary control and allow for precise, rapid movement. Cardiac muscle cells are short, branched, and striated. They're involuntary but highly coordinated for continuous pumping. Smooth muscle fibers are small, spindle-shaped, and non-striated. They contract involuntarily, often slowly, to move substances through organs and vessels. Recognizing these differences is especially important in histology labs. Being able to identify a tissue slice by whether it's striated, branched, or smooth can quickly tell you its function and location in the body. Skeletal muscle does far more than move bones. It allows us to perform voluntary movements like walking, lifting, and riding, but it also stabilizes posture by constantly opposing gravity. Skeletal muscles protect delicate internal organs, especially in the abdomen and pelvis, by acting as a muscular shield. They stabilize joints, helping prevent dislocations and injuries, and they control openings of the body, everything from swallowing to controlling urination. Perhaps one of its most underappreciated functions is thermogenesis. Skeletal muscles generate heat during contraction, which is vital for maintaining body temperature. Shivering, for example, is just rapid involuntary muscle contraction designed to warm you up. Each skeletal muscle is an organ in its own right, made of multiple tissues working together. The muscle fibers themselves are wrapped and organized by three layers of connective tissue, known as the myscia. The epimysium surrounds the entire muscle, the perimysium bundles fibers into fascicles, and the endomysium wraps individual fibers. These layers transmit force, provide structure, and give pathways for nerves and blood vessels to penetrate the tissue. Blood vessels bring oxygen and nutrients to meet the high energy demands of contraction, while nerves control when and how strongly the muscle contracts. Without this integrated organization, skeletal muscles could not perform the precise and powerful movements we rely on every day. Skeletal muscles don't work in isolation. They need strong connective tissues to transfer the force of contraction to bones. Tendons are rope-like cords of dense, regular connective tissue that anchor muscle to bone, while aponeuroses are sheet-like structures that serve the same purpose in broader regions, such as the abdominal wall. Both structures are continuous with the myscia and the periosteum of bones, ensuring smooth force transmission. A rich blood supply is essential for skeletal muscle because contraction is energy-intensive. Capillaries weave through the connective tissue layers to deliver oxygen and nutrients, while removing metabolic wastes like carbon dioxide and lactic acid. Without this vascular network, muscles would fatigue within seconds. A skeletal muscle fiber, also called a myofiber, is a single elongated cell formed during development by the fusion of embryonic myoblasts. That's why it contains multiple nuclei, usually hundreds, just beneath the sarcolemma or plasma membrane. Inside each fiber are myofibrils, long, thread-like structures that run the length of the cell. Each fiber can house thousands of myofibrils, and each myofibril contains the proteins responsible for contraction. The cytoplasm of the fiber, called the sarcoplasm, holds mitochondria, glycogen, and other components needed for energy. This highly organized design means the entire fiber can contract in unison when stimulated. At the microscopic level, contraction happens in units called sarcomeres, the repeating functional segments of myofibrils that stretch from one Z-line to the next. Sarcomeres are built from thin filaments of actin, troponin, and tropomyosin, and thick filaments of myosin. Under the microscope, these create a striped pattern. Dark A bands where thick filaments are present and light I bands where only thin filaments exist. The M line holds the thick filaments in place, while the Z line anchors the thin filaments. In the center is the H zone, which has only myosin, and the zone of overlap, where actin and myosin interact. During contraction, the filaments don't shorten. Rather, they slide past each other, pulling the Z-lines closer together and shortening the sarcomere. This sliding filament mechanism is the foundation of all muscular movement. The neuromuscular junction, NMJ, is where the nervous system meets the muscular system. A motor neuron releases the neurotransmitter acetylcholine, ACH, into the synaptic cleft, and ACH binds to receptors on the motor end plate of the muscle fiber. This triggers an action potential that spreads across the sarcolemma and dives into the fiber via T-tubules. The signal stimulates the sarcoplasmic reticulum SR to release calcium ions into the sarcoplasm. Calcium binds to troponin, causing tropomyosin to shift and reveal myosin-binding sites on actin. Myosin heads attach and pull the actin filaments, producing contraction through the sliding filament mechanism. ATP is crucial here. Not only does it power the myosin heads, but it's also needed to detach them during relaxation. The process of contraction begins when a motor neuron releases the neurotransmitter acetylcholine at the neuromuscular junction. This chemical binds to receptors on the muscle fiber's membrane, or sarcolemma, causing sodium ions to enter the cell and depolarize the membrane. The electrical signal rapidly spreads across the sarcolemma and into the T-tubules, ensuring the signal reaches deep into the fiber. This triggers the sarcoplasmic reticulum to release calcium ions into the sarcoplasm. Calcium then binds to the protein troponin, which shifts tropomyosin away from actin's myosin-binding sites. With the actin sites exposed, the conditions are set for contraction to begin. The sliding filament model explains how muscle fibers shorten during contraction. Within each sarcomere, myosin heads of thick filaments attach to actin of thin filaments, forming cross bridges. The myosin heads then pivot, pulling actin filaments toward the M line in what is called the power stroke. This action brings the Z lines closer together, shortens the I band, and leaves the A band unchanged. As countless sarcomeres shorten simultaneously, the entire muscle fiber contracts. Importantly, the filaments themselves do not change length. Rather, they slide past one another, producing the tension that allows muscles to move bones and generate force. ATP plays a central role in contraction. When a myosin head binds actin, it still carries ADP and phosphate. The release of phosphate strengthens the bond and initiates the power stroke, pulling actin inward. ADP is released, and then a new ATP molecule binds, causing myosin to detach. Hydrolysis of ATP resets the head to its cocked position for another cycle. This cycle repeats rapidly, driving contraction. ATP is also needed for calcium pumps to return ions to the sarcoplasmic reticulum. Without ATP, the cycle halts, and myosin cannot detach from actin, producing the stiffness of rigor mortis after death. Muscles require a constant supply of ATP, but stored ATP only lasts a few seconds. For immediate needs, creatine phosphate donates its phosphate to ADP, generating ATP for about 15 seconds. After this, anaerobic glycolysis provides energy by breaking down glucose without oxygen, producing 2 ATP per molecule and lactic acid as a byproduct. This fuels about 1 minute of intense activity. For sustained contraction, aerobic respiration becomes the main source, producing about 36 ATP per glucose in mitochondria. Oxygen, carried by myoglobin, supports this pathway. Aerobic training enhances circulation and oxygen use, delaying fatigue and improving endurance. Relaxation begins when motor neurons stop releasing acetylcholine, ending the signal. The sarcolemma repolarizes, calcium channels in the sarcoplasmic reticulum close, and calcium is pumped back into storage using ATP. Without calcium in the sarcoplasm, tropomyosin moves to cover actin's binding sites, preventing further cross-bridge formation. The muscle fiber loses tension and relaxes. Fatigue can set in when ATP levels fall, lactic acid lowers pH, or ion imbalances disrupt calcium flow. Over time, training can lead to hypertrophy, an increase in myofibrils and strength. Disuse or disease leads to atrophy, shrinking fibers, and reducing muscle power. To summarize, skeletal muscle is a highly organized tissue built for controlled force production. At the macroscopic level, it's an organ made up of muscle fibers, connective tissue, blood vessels, and nerves. At the microscopic level, its function is rooted in the repeating sarcomeres of each myofibril, where actin and myosin interact to create movement. The integration of nervous input at the neuromuscular junction, calcium release from the sarcoplasmic reticulum, and ATP cycling ensures that contraction is precise, efficient, and reversible. This layered organization, from whole muscle to molecular proteins, explains why skeletal muscle can perform such a wide range of tasks, from delicate handwriting to powerful weightlifting. Skeletal muscles generate movement by pulling on tendons, which are dense, regular connective tissues that connect muscle to bone. The tendon fibers are continuous with the periosteum of the bone, creating a seamless connection for force transfer. The muscle's origin is its more stable, usually proximal attachment point, while the insertion is the more movable, usually distal site. During contraction, the insertion moves toward the origin, generating movement at joints. Not all skeletal muscles attach to bone. Some insert into skin, like facial muscles that create expressions, or into sphincters that control body openings. This structural arrangement ensures that every contraction translates into useful motion or control. Muscles work in teams, not isolation. The agonist, or prime mover, is the muscle primarily responsible for producing a movement. The antagonist does the opposite action, allowing for controlled motion and preventing injury. For example, when the biceps brachii flexes the elbow, the triceps brachii acts as the antagonist to extend it back. Synergists assist agonists, adding extra force or refining motion, while a special type called a fixator stabilizes the origin of the agonist so that the movement is efficient. Roles aren't fixed. Depending on the movement, a muscle may act as an agonist in one case and as an antagonist in another. This coordinated interplay allows movements to be smooth and precise rather than jerky. Muscle fibers are bundled into fascicles, and the way these fascicles are arranged has a direct effect on the muscle's range of motion and force production. Parallel muscles, like the sartorius, have fibers that run along the length of the muscle, allowing for long movements but less power. Fusiform muscles, such as the biceps brachii, are a variation where fibers taper at both ends. Circular muscles, like the orbicularis oris, surround openings and act like valves. Convergent muscles, such as the pectoralis major, spread out from a broad origin and converge to a single point, allowing versatility in force direction. Pennant muscles have short fibers angled toward a tendon. These pack in more fibers for greater strength. Unipinnate, bipinnate, and multipinnate arrangements, like in the deltoid, illustrate just how much variety nature uses to balance power and motion. The body's musculoskeletal system works like a collection of lever systems. Bones act as levers, joints are fulcrums, and muscle contractions provide the effort. The arrangement of these three elements determines whether movement favors force or speed. In first-class levers, like neck extension, the fulcrum sits between the load and effort, similar to a seesaw. In second-class levers, like standing on tiptoe, the load lies between the fulcrum and the effort, allowing great force but less speed. In third-class levers, such as a biceps curl, the effort is applied between the fulcrum and the load. This is the most common lever system in the human body and favors speed and range of motion rather than raw power. Evolution has prioritized agility and efficiency in movement over brute strength. The naming of muscles has deep roots in Greek and Latin terminology. Early anatomists in Greece and Rome described muscles based on their shape, location, size, or function. That's why we see names like pectoralis major for a large chest muscle, or gluteus maximus for the biggest muscle of the gluteal region. Understanding etymology isn't just trivia. It's a powerful tool that makes learning anatomy more manageable. Once you recognize the root of a term, you can often decode its meaning without memorizing it outright. For example, rectus means straight, so the rectus abdominis tells you it's a straight muscle of the abdomen. Knowing these linguistic clues turns the seemingly endless list of muscle names into a more logical system. Many muscle names are like little riddles in Latin or Greek. For example, orbicularis means circular, while longus means long. Put them together, extensor carpi radialis longus, and you have a muscle that extends the wrist, carpi, lies near the radius, and is long compared to its neighbor. These linguistic clues not only help you identify muscles, but also give you information about their structure, location, or function. Teaching students to decode these names turns memorization into problem solving. Once you understand the roots, you can work out the meaning of many new terms you encounter. Muscles are typically named using one or more criteria. They can be named by location. Temporalis lies near the temporal bone. Shape, deltoid means triangular. Size, gluteus maximus versus gluteus minimus. Direction of fibers, rectus equals straight, oblique equals angled. Number of origins, biceps equals 2 heads, triceps equals 3. Location of attachments, sternocleidomastoid attaches to sternum, clavicle, and mastoid process. Or action, flexor, extensor, adductor. Once you know these categories, identifying an unfamiliar muscle becomes like reading a short description of its anatomy and function. Muscle terminology isn't just vocabulary, it's a practical learning system. Understanding these roots helps students organize information, recognize patterns, and build connections between muscles and their roles in movement. It also mirrors how healthcare professionals communicate. Doctors, physical therapists, and athletic trainers rely on these terms daily. By mastering the naming system, you not only improve your ability to learn anatomy, but also prepare to use the same precise language in clinical and scientific contexts. Terminology, in this sense, becomes less of a memorization challenge and more of a lifelong tool.